Thank you so much for taking the time to join us today on the second episode of this vlog, where we talk about various aspects of biomanufacturing, including method transfer, engineerings, and SOP development. So, Shakar, I see that you are an expert with a lot of experience in the biofuel and bioethanol industries. Maybe a good place to start with would be if you can sort of tell us a bit about your experience.
Sure. So my PhD was in biochemical engineering. I started off with enzyme fermentations and then moved into the biofuels space. So in biofuels, I have worked with 1G ethanol. I worked with 1.5G. I worked with 2G. I've also worked a little bit with 3G ethanol. So to make that distinction, 1G is mainly your starch components to ethanol, like your corn, your beet, and sugarcane and all that, easy to convert to ethanol. And I had consulted with a lot of plants, 2G plants globally. I have been to those plants. I've seen those plants with all good intentions. It wasn't scalable. And that brings back to the comments that you have been making recently on LinkedIn about tech transfer, scalability, and profitability, which are very, very important.
Yeah. So scalability, this ties in a lot to the economics of these. I see. Okay. And you mentioned the challenges with scalability, like going from the lab bench to the larger scale. So this method transfer is something that I'm very interested in, and also around engineering runs. So I'd love to hear, I don't know if you have any stories or anecdotes, or if you could just talk more broadly about how engineering runs fit into this scaling and what some of those breakdowns might have been in this attempt.
Absolutely. Great question. So Nico, engineering runs are very, very important in tech transfer. For successful tech transfer, in my opinion, a lot of times when companies skip this step, then they have a lot of headaches down the line when they go to production scale. And like I mentioned, pre-treatment reactors were not scalable. The reason being those conditions that you need for the breakdown of those sugars, the pressure and temperature conditions, and the residence time conditions, it's very, very specific.
Yeah.
So when you go up in scale, you're not able to maintain those exact conditions in a continuous flow reactor.
Yeah.
And that's the reason if some part of your biomass goes through higher pre-treatment than the other part, which is not digested fully, then you end up with some toxic inhibitors in your fermentation mash, which can totally destroy your fermentation. So going back to your question, could engineering runs have solved this problem in this particular case? Not to a great degree, but I do agree with the fact that for any scalable process, you have to go up in steps and you have to prove it at 1,000 liter, 500 liter scale before you go to the production scale. Otherwise, you're going to have problems. I was also working with a company which they had the process which was ready for commercialization. They asked me to take it to commercial scale. And when I looked at the process, it was $9 a liter for media components. And the product was selling at $3.50 per liter. Proper SOPs, like you said, SOPs can be very vague. I have seen in my own experience, I've seen some SOPs where if you think it's understood, if you don't spell it out explicitly, CMOs don't know, because CMOs are working with many, many customers. So they don't have the time to go through everything and say, okay, since they put it there, is it understood clearly? So you have to be very explicit in stating what the parameters are, critical process parameters are, what the control ranges are, and what happens if you go outside the range. It could cost you $50,000 or $100,000, but that's money well spent.
So it's interesting thinking about engineering runs as a risk mitigation tool. So I've heard that sometimes less experienced startups, for example, or people who haven't necessarily gone through the scaling process, they might be not very interested in doing an engineering run because you see the upfront cost if it's $100,000 or $200,000. But the way that I am learning to look at this is that these engineering runs are almost like an insurance against spending much more on a larger run of a product that ends up being out of spec.
Absolutely, absolutely. I mean, like the often, the cliched saying, penny smart but dollar stupid. Engineering
Yes.
runs do cost money. Engineering runs do cost money, but
Yeah.
you're de-risking the process. You're de-risking the process, and it brings a lot of value in the long run.
Okay. And you mentioned, so it is becoming very clear that in tech transfer really is a communication work. Seems to be what it is. All of this documentation, its purpose is communication. And I've heard that within organizations, internal tech transfers are often easier because everyone within a certain company will use a certain format and a certain type of documentation, and they will already be familiar with it. Whereas if it's, for example, a startup contracting a CDMO, or from CDMO to a CMO, that's different. That seems to be where the real communication challenges are, because they might have different standards or types of documents that they use and certain like terminology. Have you seen that?
Absolutely, I've seen that quite a bit. So you're right. Internal tech transfers are somewhat easy compared to external tech transfers, and that's why I also believe that for a successful tech transfer, the initial part is the most critical part of tech transfer, which is when you have the 1 liter runs and then the 5 liter runs. Uh, you know, uh, if it's a GMP product, it doesn't necessarily have to be a complete GMP lab in an engineering run to prove the proof of concept runs. And during this phase, it is very critical to have a member of the bioprocess team actually be on site at the CMO or CDMO when the tech transfer is done, so that that's when all these questions, all these, if there's any doubts about the process itself or the sampling or the DSP, all those can be vetted out when the uh, engineer is on site.
Yes. So that you having a person in plant is important for
Absolutely.
that. So
Yeah, especially during the starting phase.
Yeah. So one question regarding that is a lot of the value of the person in plant during the tech transfer to see certain details of the process that might be qualitative or difficult to describe with words, but are relevant? Is this a lot of what the person in plant is doing, sort of watching the how-to to ensure that it was effectively transferred? I've also heard this expression that if you are a factory, you don't always want the customer to know how the sausage is made. For instance, if, I don't know, oxygen or sugar concentrations are not within those parameters, you probably know how to quickly change those parameters within your plant. But whatever changes you're making, those are also likely to have unexpected consequences on the process. And if they're not documented, that can impact other factors that that you may not know about, that the contract company might be expecting.
Absolutely. Even minor things, Nico. In my experience, I've done a lot of tech transfers in the past. Even minor things like your inoculation of your main fermenter. When you say specifically, I want 250 million cells, at the end of propagation or at the end of seed culture, then I want this ratio of transfer 2%, so that my starting cell culture is this. If they don't follow it precisely, you could have fermentations that have either an extended lag phase or a very short lag phase, and they're going too fast. If they're going too fast, then your feed rate doesn't correspond to the growth rate.
Yeah.
And then there's starvation. And then you start seeing autolysis, you start seeing foaming, you start seeing other issues.
Yeah. So these are downstream consequences.
Absolutely. These are downstream consequences. These are also yield losses that you have to take a hit on.
Okay. That's great to know. So we've covered a lot, and sort of towards the end, I'd love to kind of take a step back. I think that something that I understand through this conversation is that documentation for processes and characterization of those processes, they go hand in hand.
Absolutely.
And so I wonder if what you see as emerging technologies in this industry and what you see that might be coming out in this bioethanol space that you find interesting, exciting, or noteworthy.
Like I said, when I was working in biofuel space, Nico, I mean, 1G was well established. It's been around for 40 years. 2G, there was a buzz in between 2005, 4, 5 to 2020-ish, when a lot of plants were commissioned and then they went down. Then there was the 1.5G ethanol where you were trying to push the yield a little higher by utilization of pentose sugars. And then there was the 3G ethanol, which was using algal biomass.
Yes.
To produce ethanol.
Yeah.
There was also a thrust into that side, but I have worked in all those spaces and they all have their challenges. At this point, I don't see any challenge to starch-based industry for ethanol production, unless they have a better way of pre-treating and simultaneous pre-treatment and fermentation, like simultaneous saccharification and fermentation in your 1G industry.
Yeah.
The main problems with 2G ethanol are you produce your sugars, the whole thing takes 3 days, and by the time you put it in the tank, you already have lactic acid bacteria growing there. So you have fermentation issues.
Okay.
So you not only have pre-treatment issues where you have inhibitor problems, but you also have contamination issues, which are generally non-existent in a well-run 1G plant.
Okay.
So those are what you're dealing with. But outside of that, I think you're pretty close to stoichiometric efficiency as far as 1G is concerned. It's a very simple process. It's been studied very well. I don't see that going improving any further than it already has. So if any emerging technology has to come, it has to be with some other feedstock which is easier to pre-treat than cellulose.
Yeah.
And is also economically scalable. But before you scale it up, like I said upfront, have a good understanding of techno-economic analysis, have a good understanding of COGS, do engineering runs before you take it to your production run or put metal on the ground.
So maybe one last thing I would love to hear about, talking about techno-economic analysis. So I've heard that in Brazil, bioethanol is very popular, but I wonder in other parts of the world, like the United States, what is bioethanol used for? Is it used as a fuel, considering the cheap cost of oil?
Yes. So the concept here is ethanol burns clean, so you don't have tailpipe emissions. So when you put 15% blend into your gasoline, you're reducing your carbon footprint by 15%.
Okay, yeah. So it's for blending into gas.
Yeah, you multiply that by the millions of cars on American road, you reduce your carbon footprint by 15% by replacing 15% gas with ethanol.
Amazing. Okay, well, yeah, it's been an amazing discussion today with you, Shakar, about tech transfer in bioethanol and hearing your perspective of where the industry is going. So thank you so much for joining me today. And this will be live on LinkedIn next week.
Oh, thank you so much, Nico. Appreciate it. And also congratulations to you on all the good work and all the articles that you have written. I think it's very, very critical to do a good tech transfer.
Awesome. Thank you so much. And we'll see you soon.
Yeah, thank you. Bye.
Bye.